Welcome to my molecular biology series. Let's begin this with the molecular structure of DNA. So, the double helical model of DNA was discovered by Watson and Crick in 1953. As you can see here, the two DNA strands running antiparallelly from 5' to 3' and 3' to 5'. The diameter between them is 2 nanometer or 20 angstrom. There are major groove and minor groove. The major groove is about 22 angstrom and the minor groove is about 12 angstrom. The major groove is rich in chemical info and wider than the minor groove. Both grooves aid in sequence-specific protein binding. Now what is rise? The vertical distance between two base pairs. As you can see, these are the two base pairs and the distance between them is 0.34 nanometer or 3.4 angstrom. Now what is pitch? One complete helical turn of DNA, that is about 3.4 nanometer or 34 angstrom. Pitch is also called helical periodicity. Now the double helix model is a right-handed double helix, antiparallel strands, complementary bases which form antiparallel strands allow for stable hydrogen bonding between the complementary bases. As you can see, these are the complementary bases and these are the hydrogen bonds between them. What is the purpose of helical shape? Helical shape provides stability, compactness and genetic information storation inside the DNA. So now the components of DNA. DNA consists of deoxyribose sugar, phosphate group, and nitrogenous bases. What is the difference between ribose and deoxyribose sugar? The deoxyribose sugar lacks one oxygen atom at the carbon number 2. What is the purpose of sugar? Sugar provides structural integrity to the DNA structure. Phosphate groups. Phosphate groups links with the 5 carbon of one sugar to the third carbon of the next to form the sugar phosphate backbone. We will discuss this in further slides. Nitrogenous bases, purines and pyrimidines. Small name, large structure, that is double ring structures of adenine and guanine. Pyrimidines, large name, small structure, that is single ring structures of cytosine and thymine. The difference between nucleoside and nucleotide is simple. Nucleoside lack phosphate group. Nucleoside only contains sugar and base, while nucleotide contains sugar, base plus phosphate group. The addition of phosphate group makes nucleotide. So you can see here nucleotide is formed by phosphoric acid, sugar and base. Nucleotide formation is formed by the removal of water, one from here and one from here. The bonds between phosphoric acid and sugar is the phosphodiester bond that will be formed here between phosphoric acid and sugar, and the bond between sugar and base that will form here is the glycosidic bond. Both types of bonds are covalent bonds. So the phosphodiester linkage. Phosphodiester linkage in which the phosphoryl group between the two nucleotides has one sugar esterified through the 3-hydroxyl and a second sugar esterified through the 5-hydroxyl. Let me explain you. This is a phosphoric acid and phosphoric acid forms an ester bond with the third carbon of sugar of a nucleotide, and here the phosphoric acid forms a ester bond with the fifth carbon of a sugar of the next nucleotides. Between both nucleotides, phosphoric acid forms two ester bonds. This is the phosphodiester linkage between the third carbon and fifth carbon, and this forms the sugar phosphate backbone of the polynucleotide chain of DNA. It is a regular feature of DNA. The base pairing rules. Adenine will pair with thymine and guanine will pair with cytosine and they will form hydrogen bonds. Adenine thymine will form two hydrogen bonds, they are less stable, and guanine cytosine will form three hydrogen bonds, they are more stable. This contributes to the melting temperature and GC content stability of the DNA. Why GC content, not AT content? Because GC contains more stability due to three hydrogen bonds. So higher the GC content in DNA, higher will be the melting temperature of DNA. Now let's discuss the tautomeric forms of the bases. We have two tautomers between amino and imino and between keto and enol. But remember only amino and keto form because imino and enol form rarely exist. Adenine and cytosine exist in amino form and guanine and thymine exist in keto form. I have made a mnemonic to remember amino form. Remember them as AC and amine relationship. Remember amine as amino form and AC as adenine and cytosine. You can also remember the keto form as we can go through the GT road towards the K2 mountain. So this is all about molecular structure of DNA.